Welcome to the video series in biotechnology and molecular biology. My name is Pranav, and today we're going to be talking about transcription and translation. So on the slide, I have a brief overview of what we're going to talk about today. If you're new to these topics, don't worry if you don't know all the words on there, but if you do have some experience with this is what we're going to talk about. So what is DNA? We've all heard about it, but what is it? It codes for all your characteristics, right? Hair color, height, personality, so many different things that make you you. And there is an interplay between your genetics, your DNA, and your environment. So the idea of nature versus nurture is kind of wrong. It's both. But DNA definitely plays a big role in determining what you're like. So the basic structure of it was discovered by Watson and Crick in 1953. It's a double helix, and you can see that on the top over there. There's two strands which are coiled around each other in a double helix shape. And on the outside, you have a phosphate backbone, and there's also sugars bonded together with the phosphates. Then there are four bases: adenine, thymine, cytosine, and guanine. And the arrangement of these four bases in the middle of the DNA is what gives DNA its code. That's what makes different genes different and allows your body to express different proteins. So you have the same 46 chromosomes in humans in every single one of your cells. And on the top right, you can see that that's called a karyotype. And you don't need to worry about that too much for this event. That's more of a genetics thing, but that's what your chromosomes look like under a microscope. And on the bottom right, we have a diagram of chromatin. Chromosomes are these little bundles of DNA that are really, really tightly packed. And that's what you see in the top right in the karyotype. And also on the bottom right, at the rightmost end of that diagram, that's a chromosome, condensed chromosome. But chromosomes are actually made up of chromatin, which is the DNA associated with these proteins called histones. And that's coiled up into nucleosomes, which is coiled up into solenoid chromatin fibers, which ultimately makes chromosomes and is located in the nucleus of a eukaryotic cell. So the central dogma of molecular biology plays a very, very big role in everything we're going to talk about. It was first proposed by Crick in 1957, and it states that DNA is transcribed to RNA, which is translated to protein. So you can see that on the right-hand side over there. DNA has an arrow going back to it because it replicates itself, but then it gets transcribed into RNA, and that gets translated into protein. So DNA is replicated. Why is it replicated? Cells divide. Cells need to renew themselves in case you get a cut or something, or just as part of natural growth. To do this, they need to copy their DNA because, as I said before, the DNA in every single one of your cells is exactly the same. The basic code of base pairs of adenine, thymine, guanine, cytosine, it's the same. So that needs to be the same in the new cells that are created. Scientists knew that DNA gets replicated by your cells, but there was disagreements about exactly how this replication takes place. There were three models that were proposed, and you can see those in the top right. There was the semi-conservative model, which is where the parental DNA strand, the strand that you initially have, is separated, and then each one of those single strands has a new strand attached to it during the copying process. Then there was a conservative model, which is that the initial parental strand remains intact after copying, and a completely new double-stranded piece of DNA is created. And then there was a dispersive model, which proposed that there were pieces of new and old DNA in both strands after replication. And so I'm not going to talk about the details of the Mendelssohn-Stahl experiment, but it's good enough to know for you right now that they proved that replication is semi-conservative. And if you want, you can look at the details of that experiment online in your own time. Replication involves several protein machines is what I like to think of them as, because it really does work as a complicated system of machines and very intricate parts. You can see it on the bottom right-hand side, DNA replication. On the left, you have a little green blob that is called helicase. So that is required to unwind the double-stranded double helix of DNA, because obviously that's coiled together, and for the rest of these proteins that do replication to get in, something has to unwind that double helix. Then there's single strand binding proteins, which you can see. Those are the little gray blobs on the left-hand side. Those bind to the DNA to keep the DNA from coming back together. Because if the DNA comes back together, then the proteins aren't going to have room to fit in there and do their work. Next, we have primase, which synthesizes RNA primers. And those primers are required for DNA polymerase to bind to and then create more DNA going from there. That's just how DNA polymerase works. It needs to bind to some RNA primer and replicate the DNA from there. Then you have DNA ligase, which I should say first that there's a leading strand and a lagging strand. So on the leading strand, there's just one strand of DNA that's replicated. And you can see that in the top right. Whereas in the lagging strand, there's multiple Okazaki fragments, is what they're called, which are disconnected pieces of DNA, which are synthesized independently. And then later on, they are glued together by DNA ligase. So that's what that's for. There's some differences between prokaryotes, which are like bacteria, and eukaryotes, which are like us, in DNA replication. But the biggest one is that in prokaryotes, there's only one origin of replication. So the origin of replication is where the DNA double strand splits apart, opens up, and allows replication to proceed going in both directions, as you can see in the top right of that image. But in eukaryotes, you have multiple origins of replication. And that's because our DNA is linear and because it's longer too. All right, so here you can see DNA transcription into RNA. So RNA polymerase binds to the DNA double strand and it opens it up just like the helicase did in replication. And then as it tracks along the DNA, right, it follows the DNA, it takes nucleotide triphosphates. So those are the building blocks of RNA, and it clicks them together so that they're complementary to the DNA that it's following. So what do I mean by complementary? RNA and DNA can base pair together. So if you remember, as I said before, there's 4 DNA bases, right? Adenine, thymine, guanine, cytosine. And in RNA, there's adenine, uracil, guanine, cytosine. So the thymine is replaced with uracil. And each of these has a partner base that they base pair to. So adenine base pairs to thymine or uracil, and guanine and cytosine base pair together. That's why in the DNA double strand, you have 2 strands, right? And the bases on each side are base pairing together. So that's a strong interaction that holds them together. And what's happening here is that when you click a new nucleotide triphosphate into the growing piece of RNA, it will only click together if it's complementary to the DNA base pair that's at that location. So that ensures that you're literally just copying the DNA that's right there, or you're making an inverse copy of it, right? Because you're not including the exact bases that were in the DNA, you're including the bases that base pair with the bases in the DNA. So I kind of talked about this a little bit earlier, but here's a diagram to explain it a little bit better. So on the left, you can see the structure of DNA, and on the right, you can see the structure of RNA. And what's really interesting about RNA is that unlike DNA, it's very flexible. So DNA has a stiff double helix, right? And that gives it a lot of stability. but it's not able to bend around as much, even though it coils in chromatin, as I talked about earlier. But RNA can just like flop around a lot. And as a result of that, it can hydrogen bond with itself, which is what the base pairing I talked about earlier is. And that allows it to form some very interesting conformations, and it can do some really interesting things that we'll talk about later. Right, so the last step is RNA translation into protein. So now the DNA has been copied, and then some RNA has been made from the DNA. And now you have this RNA floating around the cell. So some RNA is good as is. It plays a role in the cell, doing some regulatory thing or some mysterious function that we haven't yet understood. But other RNAs are translated into proteins, and proteins do a lot of the work that you and I know and see ourselves doing. So here we have a ribosome tracking along a mRNA, which is the RNA that's translated into protein. And as it tracks along, these things called tRNAs, which are um, RNAs that have bent back on themselves to form these hairpins, and they have amino acids connected to them. Amino acids are the building blocks of proteins. Proteins are just a chain of amino acids in a row. And so these tRNAs can base pair with the mRNA on one end, right, at the bottom, but on the other end, at the top, they have an amino acid connected to them. So what they do is, as the ribosome goes along, these tRNAs come in, and if they're complementary to the mRNA at that location, then they click, and they allow their amino acid to join the peptide chain that the ribosome is creating. So with this, bit by bit, the peptide chain is created, and using the code of DNA/RNA, again, the RNA code has just been copied from the DNA, the right protein is created. So that's all for this video. And in the next video, I'll be talking about RNA transcriptional regulation, translational regulation. So happy learning.